Morning, everybody. I guess late morning. As many of you know, EMSL's making some fairly bold plays in autonomous automation and experimentation around phenotyping, but also even bolder than that, we have some aspirations to do autonomous experimentation. And because of that, we're fortunate to have Dr. Paul Jensen from the University of Michigan with us today, who's been somewhat of a pioneer in this field and is doing things that we aspire to do in the future. So we're super glad to have you here today to give a presentation and visit with us. So Paul is an upper Midwest guy. So he went to the University of Minnesota, and I guess mostly because there isn't a lot to do, he got two majors, chemical engineering and biomedical engineering. And then he went on to graduate school at the University of Virginia and completed PhD in biomedical engineering. So I guess you made some sort of choice there. Had a great experience there, as I understand now. And then went on to the University of Virginia for a postdoc, and then from there to Urbana-Champaign as an assistant professor for a stint, and now obviously at University of Michigan. So that's all super great, but he's also an Eagle Scout. I just wanted you all to know that. That's important to me and to Paul. So Paul, the stage is yours.
Thank you very much. Thanks for the invitation and for coming. This is not my first trip to PNNL. So when I was a grad student, we had a small collaboration, and I was able to come out here and visit once before. My best friend is from Richland. So he worked at PNNL in high school, and now he teaches at Whitman College in Walla Walla. So I've been to the Tri-Cities a couple times. I only learned on this trip that you have rattlesnakes and other things that could kill me. So
they never told me about this. Nice.
Reintroduce the song.
All right, I'm going to do things a little different today because I know that people here that are very interested in automated experiments. So I'm going to try and give the talk that I wish someone would have given me 4 or 5 years ago when we started this, because we're not the first one to make all of these mistakes, but our hope is that we'll be the last one to do it as well. So I'm going to fill you in. I'm going to start and talk about why we possibly need more data. And then this idea of automated science. We'll do one sort of case study, which we call Back to AI. And then I'm going to finish with about 4 different lessons that I think are really important from people. We're going to do either automated experiments or complete closed loop automated science, right? And as I think it was Groucho Marx said, if you don't like, these are my principles, and if you don't like them, I have others. So I'm a reformed computational biologist, I would say, meaning that I started doing computational models in graduate school, and I realized pretty early on that the best thing that I could possibly do to improve my models is not some new algorithm or some new technique for the area I was working on. I was always just short of data. So I decided that data collection is going to be critical in making our models better. And the reason for this is because there are 3 things that you need in order to make a computational model go. So you need a model that is flexible enough and powerful enough and has enough mechanism or insights in it to be able to predict the thing that you want to predict. Then you need some compute power. You have to be able to train the model, fit it to data, simulate the model, and you need some sort of data, experimental results or theoretical results that it gets fit to. Now, the relative contribution of these 3 changes by field. So some fields we have very good models, but we have no data. Some fields we have perfectly fine models, we just can't compute them. We can't simulate all atom protein dynamics, that sort of thing. In my area, so doing computational systems biology, largely metabolism, I think there's sort of a dose response that looks like this. So everything we can improve the models, put more money, more effort into making better models, more computing power, more data, and we'll get some sort of return. I think the return always looks like an S shape. There's a place where it really takes off, but then there's diminishing returns, and both on the start and on the end. So the question is, where are you on this S curve for your particular field. And in my area, I think for compute power, we're already plateaued out. So we don't need bigger computers. Everything I can show you today was done on a single, rather beefy, but one desktop computer. So compute power is not an issue in the area that I work in. We also have models, and I think we're sort of exiting the exponential growth on this. So for what I want to predict, we have very good machine learning models now. I think microbial metabolism is very complicated, but it's not as complicated as the English language. And if we have models that can learn English, I think they can learn the type of things that I'm interested in. But for data, we're down here. So I really don't think we have anywhere near the amount or the type of data that we need in order to train our models. So when deciding how our research group is going to spend our effort, so most of our effort has to be in data acquisition, because that's the only place we're going to get the big return. And this is, I think, one of the only equations in the whole talk, by the way. Anyway, so the reason we're pre-boom on data is that I believe that microbiology is a data drought. And so we talk about big data and we're all drowning in data. I think in microbiology, we have the opposite. We do not have big data. We have small data. So our group quantified this. This is an analysis we did where we pulled every single paper that's ever been published on PubMed about microbes. And we took every paper and assigned it to what species does it study. And so there are, depending on what database you use, 43,000 different species of bacteria that we've sequenced and that we know is a species. Half of all the papers that have ever been published in microbiology only talk about 12 species. E. coli is number 1, Staphylococcus aureus is number 2. But it's those 12. And 90% of all the papers is less than 1% of the species that we know exist talk about it. 3/4 of the species never published a single paper. So even if you said there was going to be some AI that's going to read all of the literature and then make these startling insights about microbes, there's nothing to read about 3/4 of all the microbes that we know about. And there's a huge data drought. And I might say, well, this is great because there's too many papers and the number of papers is exploding. So this problem will eventually be solved. And that's not true. If you normalize the output of papers by the growth of the number of species that were identified, it's going the wrong way. So over the last decade, the amount published per species has decreased by 30%. So we're having an ignorance explosion. We know less and less about more and more species. So something's got to change. Right. The other issue is that a lot of people don't want to focus on collecting data. Right? Especially a lot of modelers don't want to focus. We're not the only ones that realize this. This was a paper that was published by a group out of Google. And this is a quote from one of the people they interviewed, is that everyone wants to work on the models. Your career depends on better your algorithm, that sort of thing. No one wants to sit and curate large data sets that other people are going to use. And this was Google found this. With all of our resources, we can't pay people to sit and curate good quality data sets, which is why we all train our models. Data set. Fascinating paper. I see the same thing. No grad student wants to curate next model. They want to curate data set. They want to come up with the next predictive. Okay, so we're short on data. And so our solution was to turn to automation. So we need to produce large quantities of data, exactly the data we want, and we want it on demand. And I think the way we started doing this and the way everyone starts doing this, I think is backwards. So this is my hot take part of the talk. This is me saying, I think we went sort of the wrong way. So let's explain. Scientific discovery goes like this. So we have some sort of prior knowledge. We design an experiment. That experiment produces data, and then we create new knowledge from it. That's like grad school in one line. If you can do that, you're good. Now, this is fortunately always been closed loop. Scientific discovery is closed loop because the new knowledge we produce becomes prior knowledge for the next project. So you just run in a cycle. You produce new knowledge and then update your beliefs and you produce a new one as well. Okay, so that I don't think is too controversial. That part's still coming. So the reason that this is great, like why you need this to run in a closed loop, can be illustrated using Wordles. So this is the New York Times now Wordle. How many people have done a Wordle? Okay, so the way this works, I'm not very good at it, but you guess a 5-letter word, And then it tells you if the box is yellow, it says that letter appears in the word, but it's in the wrong spot. And if it's green, it says that letter is correct. It's in the right spot and that is the right letter. And you get 5 guesses. Now, Wordles are completely solved. So a group of computer scientists out of MIT proved that with 6 guesses, how many guesses did you get for Wordle? 6.
6.
With 6 guesses, you can get to every single word in the English language if you use this optimal strategy. So there's no point in playing Wordle. I hate to ruin that. It's the problem is solved. Checkers is also solved. Checkers is a draw. Checkers, that was solved in 2008. Wordle was solved right after it came out. Okay, so you can get it, but think about that. So if you just told someone, you have to give me, I have a word, a 5-letter word, write down 6 guesses and I'll tell you if it's right. It'd be a completely impossible game to just pick 6 guesses unless you do it in a closed loop. You get feedback that allows you to design the next experiment. So same number of guesses, same problems. If you do it iteratively, it's trivial for computers. If you do it all in one batch, it's almost completely impossible. You're just taking 6 guesses. This is why we need the closed loop type of experiments. What's interesting is who's actually doing the experiments. So in traditional science, humans do all 3 parts of this. We design experiments, run the experiments, and then we turn that into knowledge. So the first thing that people did with automation is to say the problem is that humans are spending too much time in the lab and we should replace the humans with robots. And this is called laboratory automation. So a human still designs the experiment, a robot now produces data, and then humans have to sort out the data and turn it back into knowledge. Robots don't make knowledge. That's sort of a human thing. The problem is that we have now very expensive robots that wait for humans to tell them what to do. My wife used to work in a big pharma company, and she shared an office with the person who was in charge of high throughput screening for her oncology division. And I was really excited because I could go tour the robots. And he took me on a tour and he went down and he walked into the room and he turned the lights on. And they were just all sitting there. And I said, well, when do they move? And he said, oh, well, we can't run them until we finish interpreting the data from the last screen. And then we can design a new screen to use on the robots. The robots should sit there waiting for humans to tell them what to do. I visited another place and they had a big robotic system and they had a computer monitor playing a video of the robots moving. And why is the video of the robots? He says, well, it's actually pretty rare to catch the robots in action. And when people come on tours, they want to see it move. So we have a video played in closed loop as well. I guess that's technically closed loop science. They switched it into robots. Yeah.
This is my fault. That's every.
The path was so clear.
If you want the area clean, you just a really important time.
All the adjustments back.
Blowing to be done.
It's a real plan.
I appreciate all of the help in cleaning, but not seriously. Every time. So now you're good.
I think what this did is just replace, expensive humans with the expensive robot. And in certain cases, clinical automation, we're doing the same thing time and time again. This can be a huge money and time saver, but a lot of scientific discovery, this is really not bottleneck. And this was quantified. There was a group that studied a bunch of biofoundries that were set up in the UK. And they interviewed all of the people as they moved from working, doing synthetic biology without automation to automation. And it's a fascinating article that sort of chronicles their journey. And if you ask people, these are people who work in an automated biofoundry, how they spend most of their time. And the top answer is designing experiments. I spend my time designing experiments, and foundries don't change this. They do not change or get rid of the need for experimental design. That bottleneck remains. So the next thing people sort of did is say, well, if we have robots that produce large amounts of data, we're going to use machine learning to take those data and automatically glean insight. We're going to remove the human from that state. And I think this could be very fruitful. We have large quantities of data. We have not exhausted all the insight that can be gleaned from doing this. I think the issue is that, one, the pipelines are not closed loop. So we still wait on humans to design the experiments to put in and to produce the data. And in a lot of areas, like in corners of microbiology, we have no data to start this pipeline. And the second is that how humans design experiments. So mining data that already exists is not efficient for machine learning. AI and machine learning algorithms like a particular type of data. They want to vary everything all the time, and humans vary a single thing at a time, which isn't very useful for machines to learn. Right? As an example, we call this the law of bounded estimability. I don't know what the actual name is for the law, so if anyone knows what this is called, please let me know and I'll update my slide. But the order of the interactions, if you don't want to look at pairwise interactions or three-way interactions, that's bounded by the number of things you simultaneously varied in your data set. I had a colleague come to my office one time and said, I've got a big data set where we went in and mutated every single base in a sequence, and then we measured how well Cas9 can come in and cleave that sequence. And now we want to predict what's the optimal combination of these mutations in order to get the best cleavage. And I said, you can't do that. That's impossible. He said, no. That's where the machine learning comes in. You're going to fix it. And I said, if you don't have a biophysical model, if you don't have some other data set that can give you something about the interactions, there's no way you can tell anything about how two mutations are going to work if you measure them one at a time. It's theoretically impossible to do that. And a lot of our data is produced by very carefully varying one factor at a time, and we can't learn interactions from it. Okay, so instead, we need automated science. So we want to run this in a closed loop. We have to get humans completely out of the loop. How are we going to do this? And the answer is we cheat. It's the way to pull this up. So you don't create new knowledge and then run prior knowledge. We just need to keep the robots busy. So I think most automated science does this trick where we just short circuit this cycle. So we take data that's produced and we use it to train a model. A very simple model is in many cases a non-interpretable model, but that model's only job is to let an AI agent design new experiments. And when we do that, we can go right from data back to the next experiment for the robots to run, and we never have to ask a human anything. Okay, so we're going to run this sort of short circuit where we use a surrogate model to handle the experimental design problem. Then afterwards, we can take that model or we can take the data that's produced and we can do some machine learning to create the knowledge, right? But this is the way we get it to run faster, is by not replacing the humans, but just cutting them out of the cycle, short casing them. This all works because designing experiments to improve a model, asking what would be the next best experiment that would give the largest gain in the model's performance, that would reduce the uncertainty in the predictions of the model. That is not an easy question, but it's a very well-defined question. We have good algorithms that can do it. But asking what's the next best experiment that would contribute to human knowledge of a system is very nebulous and very poorly posed, and we don't have answers for that. So we're swapping out a better question, a more precise question for the one that's poorly defined in order to get this to run. Okay, so let's go through an example of seeing the short circuit in practice. We're going to talk about a couple of different bacteria that only live on the surface of your teeth. They appeared as species about when we invented agriculture. That's when we had enough carbohydrates in our mouth to support them. Normally, you have a nice, healthy, diverse group of bacteria, largely streptococci, to live on your teeth. Every once in a while, Streptococcus mutans shows up. This is bad. It causes an infection. It ferments sugar into lactic acid. The lactic acid gets so concentrated that it demineralizes your teeth and you get a cavity. That's what causes cavities. To help in this process, it forms a biofilm over your teeth. That's plaque or tartar. That's what you're scraping off with your toothbrush every night. You have a biofilm inside your jaw. Now, most of the microbes in the oral microbiome, there are about 600, are completely unstudied. We don't know anything about them. So we wanted to learn what makes these things grow. Metabolism is directly linked to pathogenicity in these bacteria. And the first thing we did is we built a mechanistic model. So this is the model we built. This is a genome scale metabolic model of Streptococcus mutans. Every reaction that we could find in the genome is put in here. It took about 1.5 years to assemble this. It took a month just to draw this pathway map of the model. Actually, it's used by a dental school in Utah to teach metabolism to dental students. They studied one of the figures in our paper. And then we decided to benchmark. So we collected a bunch of data. We said it's pretty good at predicting whether or not it can grow on a carbohydrate, but How about amino acids? So these are data showing pairwise removals of amino acids from a defined media. So these bacteria require about 40 different ingredients to define media. Kids that their parents cook for them, they never learned how to produce their own food when they go off to college. They're the same way. We feed them in our mouths, so they develop a lot of oxytrophies. So you can see that every species of bacteria, they all only exist in the same environmental niche in your mouth. They all have different patterns of oxytrophy. Black means it can't grow, white means that it grows just fine when you take out pairs of amino acids. And this is under aerobic conditions, under 5% CO2. When you roam under anaerobic conditions, you get similar oxytrophies, but there are some notable differences. So even the environment shifts the oxytrophy patterns of these bacteria. And these are all experimental data. The reason they're experimental data is that the model can't predict any of this. So the model says that these bacteria have complete biosynthetic pathways for all 20 proteinogenic amino acids. Right, so the model says you don't have any oxytropies whatsoever. We went in to try and find, is there something with these pathways? Are they pseudogenes? Are there SNPs? We can't find any reason why they can't make these amino acids on their own, other than a regulatory system that we don't know about that's shutting off the capability. So the models can't predict it. We can't find a way to predict it from the genome. Only by measuring it can we observe these complex patterns. And this is only 2 levels down. Right, so we can't even predict the leave 2 out patterns. There's a million combinations of amino acids. We can't predict any of those other than it grows when you give them all the amino acids and it doesn't grow when you take them all away. So instead, when we want to map out these oxytropies, we decided to turn to reinforcement learning. This is a technique that had taken off in gameplay. It learned how to beat chess and Go and all these board games. And the reason we picked it is because you don't need any knowledge to start. Learn how to play chess. These agents start making random moves on the board, and sometimes they win the games. Usually they lose the game, and they eventually start to associate, when I made this move in this situation, that turned out down the road to me the game. And they can develop superhuman levels of ability just beginning from scratch, random moves and no knowledge. So this is perfect because we really don't have any prior knowledge about these bacteria to begin with, so we can start from scratch. Now, normally in reinforcement learning, you have an agent that has some sort of model. It picks an experiment, something that it wants to do. It moves a piece on the chessboard, and then there's a reward signal that comes back. In chess, they actually wait till the end of the game to give the reward. Say, did you win or did you lose? They don't tell it, oh, that was a good move or bad move. They wait. It's only win or loss. So we thought we can do the same thing in the lab. So we can build a model, we can request experiments, and then we can just wait a day and get the growth data back and then feed it to the model and say, here's the reward or not. Now, the tricky thing is that the agent doesn't actually know that it's kicking off experiments in the lab. It thinks it's playing a game. It thinks it's moving chess pieces or just changing amino acids up and down. It doesn't know that this actually leads to automated experiments happen. So it comes up with an experimental design that gets sent to a machine that schedules this out and tells the robots what they need to do. We go through a set of robots that just set up the plates that contain the different reagents. Then they go to a different class of robots that do the combinatorial dispensing into the multi-well plates. We inoculate with the bacteria or some sort of bulk common media. It goes into some robotic incubators that monitor their growth over time. All that data then is sent back, and all the agent sees is what the growth is. So just the final yield of growth. We also now can do this with time series growth as well. It gets a signal back. It doesn't have to be growth. It could learn whatever output we measured. It doesn't have to be amino acids. It could be whatever inputs you want to modulate. It really has no knowledge whatsoever of what it's doing. We started doing about 300 experiments a day. Now our sort of one-day peak is we've done up to 10,000 growth assays. So these are all planned by the agent. We don't know every morning when we come into the lab what experiments we're going to have to run, and every experiment is different. We're at sort of the whims of the agent to choose the experiments, and the robots have to respond. I want to point out the tricky part on the logistics of this is that we're doing combinatorial work, but what we call high combinatorial, not low combinatorial screening. So usually combinatorial screening brings up drugs. We're going to look at pairs of molecules and see if they synergize against the target. That works, but it doesn't involve a huge number of assays, and physically you're transferring small amounts of compound into a larger well. Okay, for our case, we're doing these media screens where we're building up the entire defined media for the bacteria. So we'll put in 48 of the 50 ingredients in order to leave 2 of them out. It's a lot more pipetting steps. We have to worry about all these solvent effects because we're building up the entire media. So that 10,000 experiments could easily be 300,000 or 400,000 liquid handling operations that happen in a day. We have to have special robots because if our robots use pipette tips, we would burn through the whole project budget in a single day just buying tips. Okay, so let's watch the agent learn over time. I'm going to show you a time series of what happens. Every circle here is an experiment that was chosen by the agent. If it's a filled circle, the agent says, I think this experiment will grow in this media. If it's an open circle, it says, I don't think it will grow. We try and get the agent to predict it grows every day. The axis here shows how many amino acids it left in the experiment. So, or how many it removes. So 0 is full media, 20, it would take all already knows the answers to those 2 points. And then once the data come back in, the experimental results are in, we can say, was the agent correct or not? So if it was correct, we put a circle black and red, says it was incorrect. So on day 1, it has no idea what it's doing. It has no prior knowledge, and it gets at least half of the guess. It's just guessing. This is if you're curious, it's a randomly initialized neural network that we're using to make predictions, and then we interpret the predictions without any training as if they were meaningful, even though it's just nonsense coming out of the model. Okay. The agent is not allowed to repeat experiments. So the easiest way for it to win points is to get the answer and then send the same experiment back out again. Now, every day it has to pick an experiment that it hasn't tested before. Okay, so that's day 1, largely nonsense. By day 3, we see the number of red circles has decreased substantially. So after 2 rounds of experiments, it can pick 336 experiments every day. The agent's already doing better. This isn't a monotonic process. Every once in a while, it goes backwards. This happens usually about year 3 of grad school when it's like, oh, I don't really understand the system as well as I thought we did. There's a little more complexity here, but then the agents recover. I'm showing you every other day here, but it gets results every single day. And by day 11 to 13, it's over 90% accurate. So it can tell us the result of the experiment before it actually runs the experiment. There's no reason to continue doing actual experiments, and we tell the agent that it can stop. Okay, so no humans were involved in any of the decisions in that loop. What it's actually doing, if you visualize the phenotypic space, there's this one experiment where it has all the amino acids at the top and one at the very bottom where it has 0 amino acids. Somewhere in between is something we call the growth front, which is an imaginary line that separates the conditions where it grows and the conditions where it doesn't. And we tell the agent, I want you to find experiments that straddle that growth front, where it grows in a media, but if you take out a single amino acid, then it no longer grows anymore. So we think if we can map the growth front, we can tell if you're on one side or the other and make the prediction. So it does this in 2 ways. One is a very sophisticated policy where we just randomly take a walk through the space until it hits what the model thinks is the growth rate. This is called exploring for the agent. It's very important. The second way is we tell it to be really smart. Find the smallest media that is untested that you still think grows. That lies right on the growth front. And it has a technique called rollout where it can go and navigate this big combinatorial space in order to find that location. But it does both. Every round, it does some random experiments and it does some, we think we really know what's going on. It's called exploiting and reinforcement. And it picks its batch. A bunch of agents go out, they all find their experiments, they come back and sort of bid who's got the best experiment and who's going to fit into the batch of 336. If you cracked open the agent, inside is a neural net that we're training. And this just says, given these media, given these 20 amino acids, what's the growth that you would see as the output, right? And On day one, so this is the output of the neural network, and you see it's again just nonsense because this is an untrained neural network. So every black dot is an experimental result, a yield, a growth yield in that particular media, and the blue dot is the model prediction. So if the model was perfectly accurate, all of the black dots would be right on top of the blue dots, and you see that none of the black dots are on top of the blue line, and the blue line is right about 0.3, which is the random value that just comes out of the neural network when you don't give it any training data. Right? If you picked a new randomly initialized neural network, it would give different predictions, but it doesn't matter. We're just using it to make random starts. Now you can see the predictions of the neural net. Again, every point here is a different experiment. It's not a growth curve where these are different times. And you see by day 11 now, we can not only say, does it grow or does it not grow? We can tell you pretty much exactly how well you grow. And eventually we get, by day 13, we're within the experimental variability. So the black dots are not right on top, but if you repeat the experiment, they grow to a different final optical density. You really can't get any better than that. So now we have an agent that knows. So the neural network can predict growth, which is great, but we would really like to know. If you tell me a media, I can tell you exactly if it's going to grow, but we'd like to interpret that for the human. So we're going to do the second part. We ran these in the closed loop by short circuiting it. So just make your model better and better, give good predictions. And now we want to stop and say, all right, tell us something to humans that we can figure out. So we have a second machine learning pipeline that just takes the data and tries to build simple logical rules to explain these oxytrophy patterns. And now this is entirely by human choice. How we define complex and how we set this up is just a decision made by a human to give us something that we think that we can interpret pretty well. So if you ask a graduate student in the lab, Keenan, who built that metabolic model, we gave him the result of about 2,700 experiments that were from that first data set I showed you of all the leave 2 outs and leave 1 outs and even some leave 3 out data. It's a very small number of the million possible combinations. And Keenan sat down and poured through the data originally when he was working on his model. And this is the rule that he came up with. It needs one of the ILV amino acids, it needs glutamate or glutamine, and it needs leucine or proline. That's the result. If you ask Dr. AI, that was Dr. AI's results. This is Keenan's result on the other side. So he came up with a very similar rule without seeing the Dr. AI data, but he didn't have this leucine or proline data, oxytrophy on the end. He says you need an ILV and you need glutamate or glutamine. So his rule is simpler. If you take the 422 experiments that Keenan looked at and you score the two rules, Keenan won. The human-derived rule is better. If you score it using a randomly generated independent set of growth data, then Dr. AI wins because of that little addition. This oxytrophy for leucine or proline only shows up in very small media where you don't have a lot of other amino acids. So it's not that Keenan missed it. It just wasn't in the data set. His is more consistent with the data he had. If you look at a full data set, now this third oxytrophy pops up that Keenan couldn't have recovered because he didn't have the data. He couldn't wrap his head around those really large sets.
What was the level of effort difference? I mean, how long did it take for us to pour through 422?
So he had been working on this for a couple months, trying to develop an algorithm to fix it. So we didn't lock him in a room to say, like, go and come up with the rules. So I can't really say how long. But he spent a lot of time trying to reconcile the model's predictions and say, can we fit the model in order to match the experimental data? And from spending time with all the data, this is what he came up with. The Dr. AI, this takes about 10 minutes to come up with, after we have the data, to come up with the rule. So advantage we didn't really see is that, well, you think you have enough data to predict what the oxytropies are, but you're probably just missing a lot. Even this isn't even accounting like, well, in anaerobic conditions, we have different rules for that. This is looking at anaerobic conditions, just the amino acids. Strange oxytropies appear with smaller numbers of amino acids versus larger ones. So 13 days, we probably could have stopped after about 11 days with our previous model. And I thought that was pretty good. But it seems like, all right, we need then 2 weeks and there's 600 bacteria in the microbiome. We could probably run some in parallel. And we're trying to estimate how many this is going to take. Then we decided, well, maybe we can speed this up if we reuse some of our models. So they are similar. They're not completely different bacteria. So we learned a different model for Streptococcus sanguinis. It took 7 days to learn that one. This one was a little bit easier for Bacteriaceae. There's always some stochastic start random experiments. And then we were going to learn a model for Streptococcus gordonii. We said, rather than starting with random experiments, why don't you take the model that you finished with Strep sanguinis and use that to plan the first day. And then after the first day, just throw it away and use the data that you've collected. But it then might be better than starting from randomly. Just that one change using the previous model dropped it from needing 7 days to about 4 days in order to do the retraining and a lot fewer experiments. And so there's some value doing this transfer learning, as the machine learning people refer to it, reusing the model, even though we know that the model is going to be wrong. We also said, well, this was good for learning amino acids. As I said before, there's about 40 ingredients to this media. What about all the rest? Adding another 19 ingredients would increase the size of the problem by 500,000 fold. Right? So there's an exponential explosion. What type of scaling are we going to experience? So we started with a model of amino acids. We used that model as a starting point and then did more experiments on full media, 39 different ingredients. And it just took another 7 days to almost double the number of inputs, even though there's an exponential increase in complexity. And we've seen this with lots of other cases, that we can learn with scaling that's about linear in the input problem size, even though the output space is combinatorially large. So double the number of input variables, it probably takes about twice as many experiments, even though we're learning all the interactions among those variables. Okay, so the Bacteriaceae was focused on, does it grow or not given the media? And actually, we learned how well does it grow. We learned a continuous measure, even though we scored it using these binary outcomes. We still have these models that we know don't predict very well. And now we actually have enough data that we can go back in and fix the models using the experimental data that we have. So this is a grad student, Kurt, in the lab. So he started with a standard constraint-based metabolic model. This is the type of modeling I did as a graduate student, where we give it nutrients and we give it a genotypic state, and it can say whether or not it grows. And on the bottom here, I'm showing you the results across several thousand conditions where we have different media and the fitness that we measured versus the prediction from the model. The model's predictions here are the yellowish blocks, and constraint-based models have this sort of jump discontinuities. They kind of predict binary outcomes. They're not real great at giving you continuous measures before. So we added now a neural network that simulates the regulatory environment inside the cell. So we know that these bacteria have transcriptional regulation that is keeping them from using their full metabolic potential. So what if we take the nutrients in the environment to put it into a neural net, and the output of the neural net are adjustments that are made to this constraint-based model? It has to go in and tweak variables and learn what genes should be on or off or up or down in each one of these conditions. And there's a couple math tricks that you need, but you can train this neural network by passing information in and out of the constraint-based model. And you can now fit the parameters of the neural network to your experimental data, and you get results where we can predict things better. We get more continuous level outcomes. These are some early work. The predictions have gotten a lot better since we've improved the training technique. But now we have constraint-based mechanistic models that are asking for experiments. What would be good for updating this regulatory network? And we still have some mechanism. We don't know what the transcription factors are, but we know that when we change an environment, that this is the result. It's up or down regulating these pathways inside the cell. How it's doing that is still a black box, but we can get an idea of where this information is targeted. This is only with phenotypic data. Have some genetic data now we can fold in, but there's no ChIP-seq, there's no RNA-seq used to train this. It's only on the growth assay. Another grad student in the lab has started to work on dynamics. So given an input, this is what's in the media, can you not predict, is it going to grow, but can you actually predict the growth dynamics, get the full growth curve of the model? He uses something called a neural differential equation. So we have a neural network that's embedded inside a system of differential equations that learns the dynamics of the system. So the monad kinetics, the growth kinetics that go on inside, that's what we're training. Again, using time course data, this is one result that came out. So this is a prediction. We tell it, this has 8 different carbohydrate sources in this experiment. So we tell it the concentration of all 8 sugars at the start, and we tell it at the OD that it's inoculated at. And then you see the actual measured growth dynamics and the predicted growth dynamics. Everything after the initial time point is all predicted by the model. You see the jump in the middle there, there's a discontinuity. So we actually pass the bacteria. We knock down the OD and we change the concentrations of the sugars. Right? So we can tell it when it's doing the forward prediction at time, whatever we're going to put in, we're going to dilute the OD and we're going to put in this amount of these sugars, and the differential equation can respond to that discontinuity and jump as well. So we can learn dynamics rather than just endpoint measurements. The last goal is that we've been viewing sort of the cell as this input-output factory black box. We now have a research faculty in the lab, Ryan, who's led our campaign to learn the mapping from genes to phenotype as well. So he developed this really elegant system to do genetic perturbations. So we've been producing at the rate of about 600 strains a day. These are knockout strains, overexpression strains, underexpression strains, fluorescent reporter strains for these bacteria. So we're going to have genome-wide libraries of all these genetic perturbations that can now be fed to the robot. The robot can say, I want you to grow it in this media, I want you to delete this gene, and I want you to measure the expression of this protein. And be able to learn the coupling between genotype and phenotype as well. So this we're excited about. Today, I think they're just starting the library. They've done the knockouts, but they're doing the first batch of strains. So it'll take about 2 or 3 weeks to make reporters for every gene in one of these species of streptococci. Okay, before I get into the lessons, the big misconception, I think, when people talk about automated science is that originally science was done this way, where again, we have humans that design experiments and humans interpret the data and it runs in a loop. And what I've talked about today is this way that we call automated science, where an AI plans the experiments and then the experiments are run by robots. So there's no human involved whatsoever. And people usually think that it's one of these two extremes. Either humans are going to do science or there will be no humans doing science. And I really don't want to spread that impression that there are things in between. So lab automation, as you already mentioned, is you can have humans plan experiments and then robots can carry them out. And this is what a lot of automation facilities will do for you. The other one that we should all be aware of is what we call model-guided science, where you still use the AI to plan the experiments, but these experiments are really difficult to automate. They're very expensive. We don't have nice things we can buy that'll do the liquid handling or glycerol, which we can't pipette very well to the robot. Whatever the reason, you can't automate the experiment. You can still have the human run the experiment. They just have to listen to the AI's plan. Right? And we've done this. We have a grad student who was able to optimize rRNA depletion for microbial bacteria. He used 31 total experiments, but he ran it in a closed loop using statistical models to tell him what conditions to do next. And he was able to decrease reagent costs and improve the efficiency of rRNA. And he didn't have to do any experimental design, but he did every one of the experiments without the robots. So don't think, we don't have an automation system, we can't start incorporating this. You absolutely can. And I think the value of this bottom corner here is really underappreciated. Okay, here are the lessons. So I have 4 lessons for you if you were going to try and do the same thing that we did through a number of mistakes. Number 1, do you really need robots? And I would tell most people, like, the answer is probably no. Most people think I have a throughput problem and say I'll use a robot, and now you have 2 problems instead of just 1. So this is our rule. This sort of law of the lab has turned out to serve us really well. Right? So if we are going to do tens of experiments for something, we just do them with single channel pipettes and Eppendorf tubes and and they proceed as normal. If you start to get to hundreds of experiments, then we don't use any robots. We just use multi-channel pipettes and multi-well plates. And that works really well. Only when you're going to do the same experiment thousands of times can you start to use the robots, even though we have all the robots in the lab, because it'll take you longer to get the robots up on a protocol than it will for if you just did the protocol by yourself, by hand. All right, so our gene knockout strains are done with robot assist. We have a couple steps that are done by robots. The rest is sort of done by humans in between because we're making thousands of strains. Once you get to tens of thousands, then go for the full automation. But I think that's the break point. Anytime before that, and you're spending too much time spinning up the automation, and it would be easier to just have the humans do it instead. All right, so I'm here to discourage people from early automation because I don't think it's worth it until you get to the end. The second one is realize that there's nothing called lights off automation. That term came from a former undergrad who works in industry. The idea that we all go home and the robots continue doing science without us. During the pandemic, everyone said, you must be doing pretty well because you have an automated lab. So obviously science continued. Like, well, no, the robots don't work if the humans aren't there watching them, preventing an uprising, that sort of thing. So there's always a human putting the reagents in. I mean, some human has to approve the order for supplies at some point to come in. They have to clean the biohazard waste, they have to calibrate, maintain the robots. So there's a human in there. And we know there's a human in every one of our experiments. So we use the human, right? We schedule the human, the AI gives instructions for the human, right? And we've split apart for every fully automated pipeline. What are things that are easy for the human and hard for the human and easy for the robot and hard for the robot? For instance, we very rarely have robots transfer plates between machines because that is really tough and very expensive to automate. So humans care. We have technicians and the system will say, please move plate 3. It's very polite. Please move plate 3 from this robot to the other one, upload this file that I wrote for you and press run. And then we'll move it back and forth. We slowly then have added automation to a couple of our robots that are real bottlenecks. But other than that, there's human in the loop all the time looking at it. It's tough to replace them. Amazon is very good at this. They slowly automate. Right now in the Amazon warehouses, all the humans do is grab things and move them. And the robots move everything to them because grabbing is very difficult for robots. Ginkgo Bioworks did a study of all their automation failures. 80% are grabbing plates, moving plates, flips the plate, flings the plate. When I was a postdoc, we had a robot that threw an incubator across the room. So we don't do that. They're very good at pipetting. The third lesson, you need to automate QC. When we fired up this process, we can do thousands of experiments a day, which means we spend our entire day QCing thousands of experiments. Like, did this work? Should we rerun this plate? Should we not rerun this plate? All these decisions piled up and QC became the bottleneck. This is one of the robots we use. It's made by a company called Formulatrix. It dispenses nanoliter droplets using this microfluidic chip on the bottom. So it uses pressure to thump a membrane and fire a droplet out so you don't have to change any tips. You do have to pay to replace the chips after every million cycles or so. So we have a machine learning algorithm that QCs the robot. We run an assay through it that has dyes dispensed. This goes into a plate reader. The plate reader uses statistic process control in order to figure out if one of the nozzles are off. Because there's 48 nozzles on this machine, and it's going to do 250,000 dispenses during a day. And it's nanoliter droplets. So we have no way a human can tell if one of the nozzles is off or that chip needs to be replaced. And you can see these spikes. So all these lines should be the same. We're asking it to pipette the same volume every day. And every once in a while, we get a nozzle that spikes. So we need to take it out and clean it and service it. We run this before and we run this after. And if something fails, we throw out the batch. We dump the experiments. So all of our robots, they have to have this QC process. How do you judge if the data are good? We run in 384 well plates, and 48 of the wells are controls, positive and negative controls in every plate. We give a huge fraction of the plate just to control so we can detect and correct errors. So we also rerun every day 10 to 20% of the previous day's assays. And if those don't match up day to day, we throw out the batches and we go back and run it again. So a large part of our throughput is spent just on quality control. The final, probably the biggest lesson is that high throughput, which was originally our goal, should not be the objective you're going for. So I told you that we can run up to 10,000 assays a day. And then I told you that in Bacteria AI, the robot picks 336 experiments every day. Right? Because we don't need that many experiments if the experiments are well designed. The actual amount that you learn is a product of three things. So how many experiments can you run? How many days out of the year do you actually run at that throughput? How many days are the lights on? It's some of there. And then the information density of the experiments. How well planned are they? Is there a statistical or information theoretic thought that went into how the experiments were planned? And most people just focus on the throughput and build a big machine and then it runs once a month, rather than you can have a lower throughput machine that has a much greater uptime and runs every day. I think the biggest bang is that most experimental high throughput screens are very information sparse. Right? They are single, one at a time perturbations. They're screening on growth on individual nutrient sources or something. Those are not information dense and they're difficult for the models to sort out interactions from that. So if we were going to invest in improving the output of our system, we would focus on experimental design and not adding more robots until we realize, oh, now we're at optimal experimental designs and we just need to do more experiments. But most people, including us, built the opposite way. And then we have capacity that's unused because we thought we were going to get the answers through throughput. It's not the case. So my strong opinions weakly held is that a lot of corners of biology are not in the big data revolution and are far from entering the big data revolution. We still need to produce the data. Once you have the right data, but you have designed experiments for the models, then learning them is relatively easy. We have data on demand, we have unlimited data. A lot of these modeling problems, not having the right data or too little data, go away magically. And when you're designing production systems, you should sort of maximize the information that you get out of every experiment, not how many experiments can we push through every day for a given experimental budget, because that can lead to really weak data sets in the end. So with that, I want to thank lots of people who did the engineering work on this project. Bacteria AI was led by Adam Damley, who was an undergraduate. So he was finishing up undergrad and he had to stay for personal reasons in Illinois for 2 years. So he was a postbac. He was the person that integrated everything and programmed all the robots and built the machine learning models. So it is not, with no insult to Adam whatsoever, it was a wonderful, it is not a hugely complicated AI masterpiece. It's something that you, with off-the-shelf components, you can put together. Adam now works in the San Francisco Bay Area doing automation of a biotech company. So he gets the lion's share of the credit on the work, and thank funding sources and all of you. We're not trying to keep this a secret, so I would love to tell anyone about automation and high throughput experiments. I teach a 2-day workshop with my friend in Walla Walla to get companies and at academic institutions that want to start learning exactly how these models work and how they do the data collection. Our goal is to get as many people as possible doing this type of automation work. So I'm looking forward to lots of fun conversations with people here. That I'll leave you with my favorite quote, and happy to take any questions.
Thanks, Paul.
There are questions in the chat as well.
Where do we start? We're going to go with Dr. Baker first, and then who can queue up some questions in the chat? So
They're all from the same person, so
Yeah.
you could perhaps just call on that person and let them.
Sarah can let us know who that is. Thank you.
So my question is, I was intrigued by when you said you transferred the model, the endpoint model from one organism or one set of experiments to the next. Do you assume that the output of that next, after you've transferred that model as a starting point, that output would be the same if you tweaked that starting model? So if you had a whole bunch of different tweaked starting models, would you always end up at the same endpoint, or would that endpoint, could it differ, and how would it differ?
it would, I'll give the easy answer. It would definitely change how fast you get. If there is one endpoint, it would definitely change how fast you get to that endpoint. Because you can start from very bad models. I think if this were going to be deployed at scale, I think what we're going for is probably a first round from a blank slate to figure out which model is a good starting point. You know, so we have models from different bacteria that have similar, you know, E. coli and streptococcus are very different. E. coli, you need sugar and salt and you grow, and ours, you need 50 vitamins and minerals to grow. So a first round is saying which is a good model to start, that to sort of kick off the first round of experiments, and then you'd get convergence after that. We've rerun this, so we've started back again from completely random. Reinitial a brand new neural net, initial random state, and we get convergence to exactly the same oxytrophies again. Now the transfer in a new model could be a bigger perturbation where that's not the case, but we don't know. We haven't found a case where we end up at, oh, this run we got, now it seems to need algae. But there are probably phenotypes where unless you get one experiment to show that that's the case, you're going to miss it. That does keep me up at night.
Oh, thanks.
Who's our person?
Jeremy Zucker.
Jeremy,
Hi.
you're up, and then Bram's up.
Great. So first of all, Paul, this is fabulous work. I really enjoyed hearing all the progress you've made on laboratory automation. I had some very specific questions just about some of the modeling approaches that you were doing, and just how one could incorporate prior knowledge to help accelerate the training, right? Because it sounded like you started off with a very kind of model-free reinforcement learning algorithm, and then didn't know anything, and then it slowly learned the phenotypes. And I guess my question is, why not include the metabolic model in the reinforcement learning algorithm? That way, you can iteratively add and remove reactions such that the model can explain the observed growth, no growth phenotypes. So that was my first question. The second one, and then as you continued on, you talked about how you could use a reconstruct a regulatory network, and then how the network itself was learned, and you had the regulatory and you had the genome scale model. And so then my question for that situation was, well, could you combine, if you actually had a reconstructed regulatory network, could you combine that prior knowledge with a reinforcement learning algorithm to infer condition-specific constraints on the COBRA model, right? Because I think one of the points you made was a genome scale model is kind of dumb in that it assumes that all the reactions are on all the time, whereas there could be condition-specific regulation that would cause the model, it would cause the organism not to grow, and that would be something that the model might miss. And so by including regulatory constraints, a prior knowledge of the regulatory constraints, you might be able to pick those condition-specific oxytrophies a little bit better. And sorry, and the third question was, and this is, I'm trying to really understand the utility of a prediction software that gives you no insight into the model into the it gives you no insight into the metabolism, right? So my question is, if you wanted to genetically modify the organism to introduce an oxytrophy or remove an oxytrophy, then how could the reinforcement learning predictions that were trained on the unmodified organism help?
Right. Okay. I think the answer to the first part is, I think it's a good idea to use prior knowledge as some way to pick informative experiments, but I would be very wary to make that all of the basis. So I would say, yes, if you have a good model, bring it in, have it pick half of your batches of experiments, and then the other half, are some randomized or some other design. Because like in our case, we had a genome scale model, but the model predicted for every case it grew, unless you took even when you took all of them away, the model, there was no way we could have planned using that model. And that's probably an extreme case of the model being really bad. So we now have, when we're building these models that have the neural net fused to it, we do ask the model to pick the rounds of experiments, because we want experiments that decrease the uncertainty of that sort of mechanistic model. For the last part, like I said, these models are completely amechanistic. They are black boxes. And we view sort of interpretation as a separate step from data collection. So the model's value is only predicting, say, this would be a good experiment to do in the lab, even if it doesn't know why. And then afterwards, we take and distill mechanism out. We build a mechanistic model and we parameterize it. And we think there's a breakthrough point there. Eventually, our models get so good that they can predict the outcome of any experiment before we even run it. So after we get to that point, we, in essence, have infinite experiments because we don't have to do any more experiments. We can just ask the model to give us any result that we have. So what we really do is use it to say, now we've collected sufficient data. This data has the information inside of it that you would need to understand the phenotype. And then you can hand it to the modeler and say, train your model, but I know you're not going to hit limitations on, do I have the right data? You know, everyone has that. Do I need more data? Do I need a more flexible model? That issue we can solve by this sort of breakthrough where the model's trained, but you have to get the mechanism back out. I agree with you.
Okay, Bram.
Thanks. Paul, very cool talk. I think I can say I like it more than Jeremy, and by Price is Right rules, maybe that means I win. But so something your team has probably given some thought is, how do you get around some of the issues with just not being able to culture most bacterial species at all? Is this something you think could be another target for your system?
So if I really wanted to start a fight here, I would argue that I think most bacteria are culturable and I think most are transformable. That's a set. We didn't even talk about the other part that I was working on in the guy, but I'm hoping to stay till tomorrow. So I don't we have one group of students that's working on this idea of a lot of microbes from the oral microbiome you can grow if you dump in the kitchen sink. So there's one Treponema denticola. You can culture it in a defined media. That defined media has 98 ingredients in it, and it probably requires a lot fewer than that. So we're working on variants and algorithms that can say, if you dumped in a couple hundred metabolites and got this thing to grow, how quickly can you get that down to an actual defined media? Yeah. That's a difficult learning problem because it's high dimensional and the amount of media that it grows in is very, very exponentially small. But we're making some headway on that. So I think that's going to be the solution, is we eventually have the super cocktail 500 metabolites that we use as the starter media, and then the AI can very quickly turn that into something reasonable. Because I don't think the microbes need 500 ingredients. I think they just need a strange combination of 20 or 30. That's an argument for another time.
So we're going to go Luang Zhang, Javi, and then David. Going once. Here we go.
Hi, Paul. Thank you so much for this wonderful presentation. Yeah, so my name's Lenny, and I'm a computer scientist within the lab. So I want to give you some thoughts from a computer scientist perspective, right? So in your presentation, you mentioned something about transfer learning, right? You said that you leverage transfer learning to be able to achieve higher performance and higher throughput, right? To be able to run these autonomous experiments faster and to run more autonomous experiments in a certain amount of time, right? So from that perspective, right, I think you do care about the computer power and also the computer performance optimization techniques, right? So, and also within the autonomous experiments, which is a closed loop, right? I'm wondering if there are any data curation and data processing, data analytics interfaces that connects two steps, like from this device to the next device, right? If that's the case, right, you do want some better computing power that can accelerate that, right, to be able to achieve better performance and a higher throughput, right? And also in computer science, we have those lots of optimization techniques like pipeline, data reuse, and parallelization, right? Because you have this a bunch of devices that runs in sequential, and maybe one of them could be idle while the other ones are running, right? And how can you leverage the idle ones to make it working with that's to achieve a higher performance, right? That is another problem. So I want to see if you have any comments or thoughts on about of this that I talked about.
Yeah, I think we can sweep computation under the rug. So we get the experimental data back, and we actually only have an hour to do all the model retraining and the planning of the next batch of experiments before we have to start the robots. Otherwise, it moves to the next day because the robots take quite a while to do all the pipetting. So our planning and retraining model actually had to be parallelized over a multi-machine in order to do that. And I think the same, like now that our scheduling, the robots gets more sophisticated, we hit the same issue. We solve about 6 mixed integer linear programs in order to plan every batch of experiments. And we're starting to hit computational limits. I would agree with you that the more we do it, and the better we try and do it, the more computational things we do. Yeah.
Right. Okay, that's good. Yeah. So we did have similar parallelization workflow for machine learning reinforcement learning training here at PNNL, and we managed to achieve like a 1,000 times performance speed up, right? So it's from like 5 hours to 20 seconds, right? So I think that would be helpful, very useful if right, if you are interested.
Well, very much so. That'd be great.
Okay, thank you. Javi and then David in reserve on deck as it was.
Okay.
Hey, Paul. Yeah, thanks for the great talk. Lots of cool stuff, and thanks for your time and answering all these questions too. I guess my question's more of a practical one. I'm wondering if there's any way that you've thought of to test drive these models in silico without, dumping all the resources for running thousands, tens of thousands of experiments a day before figuring out how fast your model learns or is able to get to that level of predictive power you'd need for confidence in the results.
Yeah, we struggled with this. So originally we thought, we estimated this was going to take 6 to 8 months to learn the oxytrophies. And that number, we have no idea how we arrived at that number. Maybe it was in the grant application, I don't know. But we had no idea. And so we were trying, and then we would dream up these fake models. So this is what metabolism looks like, and then try and learn it to get the hyperparameters for the AI. And then we realized that it all depends on what model we dreamed up, and that's not a good thing either. So we have no idea when we finally said we got to turn it on and let it run. And we had no idea how fast it was going to work or if it was going to work. And the policy that we picked was not tuned ahead of time. It just happened to work really quickly. So now that we have models for some bacteria, we've gone back in because now we have something that's realistic. This has the oxytrophy patterns of a bacterium. And now we're working on finding better policies to say, okay, there's a smarter way to do this. And we are finding smarter ways than that. So I would not encourage anyone to implement exactly the algorithm in the paper because that was one thing that worked. It was not the optimal solution to any of these because we didn't have a model, but we do now.
Sorry.
He saw right through us. We should have done the optimization.
Well, thanks for your talk, and I just wanted you to comment maybe on a couple things of, I mean, we all share some ideals with DOE, et cetera, that we might want to have different bioproduction and bioproduction targets and so forth. And this really streamlines a lot about what's optimal for growth, but then what challenges do you see as you move away from just what's optimal for growth and the deeper understanding that's needed to say what's the most efficient way to produce a bioproduct, or if you have knockouts where you should, took a month to write up your metabolic model. If we pre-plan closing off certain parts of metabolism, where and how do you get to the most efficient model for a bioproduction, and what problems and value do you see from going from small volumes to the larger production that's needed for bioproduction?
I wish I had answered the scale-up question. That would be, I think I'd be a rich man if I knew how to predict the scale-up failures. Separation failures too, I guess. If you can measure it, we can learn it, is sort of the mantra that we've got to. So if we do fluorescence tag of a protein, and then we can watch the expression of it, and we can use the same exact algorithm to learn what is it that influences the transcriptional dynamics of this protein. We don't have the capability to measure any metabolites, which is why we focus on growth, but the algorithm doesn't really care what it is the output is, or if there's just one, you can do multi-output as well and learn that input-output relationship, as long as there's a way to give a quantitative signal of it at the end of every round. The one case where we've done this, we've worked on riboflavin production. These bacteria can't produce riboflavin, so they lack a lot of the regulation that messes up riboflavin production. So we have engineered strains now that produce riboflavin at high levels. We've done both genetic and environmental optimizations to figure that out, because riboflavin is fluorescent, so we can measure it in our flavor. We don't have to do any mass spec or NMR to measure riboflavin. So that's one case where we've been able to do it, but it's just the reason we picked that is because of our technical limitations. So I think if you can, anyone who can measure something, this is an option, and it doesn't take many experiments. Our riboflavin ones are dozens of experiments, not thousands of experiments.
So there's value in making those measurements on the scaled up volumes. And that's what you're saying.
So, and I think there'll be transfer too. Like being able to learn and do it in a sequence, maybe that will tell us about what changes on scale ups enough that we don't have to do all the intermediates. You don't have to go to 4 meter and 10 meter before 5,000 meter.
Awesome. Thank you.
We got some more questions from Jeremy Zucker as well. Jeremy, if you want to unmute.
Sure.
Hold on,
Thank you.
Jeremy, just hold on a second.
No problem.
Sorry to interrupt you. So I just want to make the point that the agenda has this going until 12:30 because we wanted to keep some open time for discussion. But many of you probably walked in the door thinking it was an hour. So for example, the director, I want to offer everybody the opportunity to leave, but we'll stay to have the conversation until 12:30 for those of you that are interested before we go to lunch. So if you need to park, go.
Well, thanks for the talk.
Yeah, and some of you have time with him later too. Lots of you have time with him later, so you can go on to other things. That's the reason to leave now. Okay, Jeremy, you're back on deck.
All right, thank you. So first of all, again, I am really impressed with what your self-driving lab can do. My question is more kind of like focusing on how we can leverage the metabolic model and comparing that with what can be obtained from a model-free reinforcement learning algorithm. And in particular, I'm concerned about a situation where you have, like a reinforcement learning algorithm can certainly predict auxotrophies that it hasn't observed yet after it's learned enough auxotrophy, learn from enough auxotrophy information. But what happens if you start to genetically modify that organism and ask what are the auxotrophies in this modified organism? And I'm concerned that it wouldn't be able to extrapolate as effectively as a genome scale model would, because it contains all that information about what genes are catalyzing which reactions, et cetera, right? Now, I guess you could still fine tune, and that I think that was your point. You showed that the transfer learning, you could fine tune a model that was learned on one strain to another strain, right?
Yeah, no, so you're right in one thing especially is that you can't with the pure reinforcement learning technique, you can't learn anything that you're not only measuring but manipulating.
Right.
You have to be able to change it so that it sees how things go. So the solution we've kind of taken when we put the model back in the reinforcement learning loop, you see, so the diagram here on the right, we have the output of the neural network predict changes to genes, and this is so that we can still do genetic perturbations. So because it's manipulating genes, and now when we grow a gene, a knockout strain of this particular enzyme, we can, despite what the neural net says, we can still make the perturbation in the model and then get the benefits of having that genotype phenotype mapping inside the model. So that we don't know anything upstream of that. So why that gene went up or down, that's now the new black box, and we would need other genetic experiments to figure that out. But we think we can recover some of it. What's interesting is that even though we're not using genetic perturbations, just having the model in, we can it can still respond to some genetic perturbations correctly. So it's learning that because we have the constraints that these enzymes are the only ways you can make the biomass products, it can sort out some of the genotype phenotype mapping, correct some of those things in the model. Yeah, I think there's advantage to, like you said, keeping the model in the RL loop and having that drive the experiments. It needs to be some kind of Bayesian as well. A lot of the RL techniques require some posterior distribution, but there's good techniques for doing that too.
Yeah, very cool.
Thanks, Jeremy. Anybody else? Ivette.
Thank you for the talk. Yes. So can you comment on what was so far the instrument that was most difficult to automate and integrate with your agent?
Yeah, the one that we didn't automate, which was plate transfer back and forth in between. We don't do plate handling around. I have lots of good information about if you're doing media combinatorial phenotyping. We bought a lot of the wrong robots that we would have gotten different robots in the end. And we have some ones that work really well for us. Not that other robots are bad, but just they don't work well for the type of pipetting we're doing, where we want to do lots of combinations of things. Most of our robots are from the crystallography divisions because they have the same idea where they like to throw all sorts of combinations in the hope that they get crystals out. So the instruments that we use are made for crystallographers, and the companies are kind of curious as to what we're doing with them. So we buy some from Formulatrix. The one we use most is by a company called Art Robbins Institutes, called the Cobra. But if you're interested in exactly what equipment we use and for different ones, I'll tell you exactly what we think we should. In total, we use about 16 different robots, detector, plate reader, type things, and that's changed over time. But every one project doesn't use all of those.
Do you have any plans to automate mass spectrometry there too, or you don't need it so far?
I don't know anything about mass spec, so I would love to work with people who can do metabolomics, proteomics, and all that. We do lots of genomics. We do transcriptomes, RNA-seq. We do phenotyping, and we do some optical microscopy. Now we started to do confocal. I can't touch the rest, and that's our. Whenever someone asks me, "What's your Achilles heel?" I say we don't do metabolomics and we don't do proteomics.
Thank you.
So how well or quickly could your model development recover from a mistake, like a a plate put in backwards that then is not detected. I would imagine it would cause a hiccup, but you might eventually end up at the same endpoint.
Yeah, just take longer.
Yeah, fun story for you. So there are 40 ingredients in the defined media. And we started with 20 of them are amino acids. And we did the first round of review while we said, we'll do the amino acids and then we're going to work on the bigger one. We'll do the other 20. And because we were terrified that it's not going to scale. We need some better algorithm. It turned out to scale, but if you read the paper, we varied 39 total ingredients instead of 40. And the reason was that we realized after that there was a bug in one of our systems, and it was outputting the wrong pipetting instructions to one of the robots. So one of the ingredients was never varied. It was always just added into the media. And we caught it because when we get the result back, it said that metabolite X is not essential. It doesn't impact growth. And we went, well, it seems like that actually we know that it does. And then we found that, the robot had been flicking this thing on and off, but it never actually happened in the experiments. And then we didn't know. So we said, well, we only varied 39 instead of the 40 ingredients, and that's why there's 39. All that's to say that, we found that by chance, that we saw that the results didn't match. Now we have a team that's working on minimizing human error, how we can stop plate dynamics. So QR codes on all the tubes and taking pictures, and to make sure that, what we're terrified is if you put alanine or glutamate instead of glutamine at a spot in the robot at the start, it's gone. All our experiments are off, and you grabbed the wrong tube off the shelf. So we're trying to document that and then look for inconsistencies in the data, basically predict surprising points given prior rounds. To say like, this is really, this result was not predictable before. Does that need to be evaluated again? I'm sure there's some.
So is that, I guess what you were describing is a systematic continuous error. I'm thinking of something that's more of a stochastic error. Is the model recoverable after, since you can't repeat the experiment,
right?
Are sometimes results, can they be ignored?
You saw one example, right? You had it started to work poorly, and it
got worse than the prediction.
Yeah.
and found itself and bounced out.
So here's the interesting thing about stochasticity. So we did, when we moved to the larger ones, we tried to figure out, what's optimal in terms of number of replicates that we should be doing? Because we do 3 to 4 replicates for every experiment, and said, can we get away with fewer? Should we be doing more? Because now we're going to be scaling up. And we found the optimal number for the model. So the model would much rather that you give it single noisy data points, but 3 times as many conditions, rather than a third of the number of conditions, but you actually have statistically average results. And this is because of Ronald Fisher developed this with the line of experiments literature. It's called hidden replication, that the model can eventually sort out the averaging if you're varying enough stuff. And we found we were wasting time on replicates. So I don't like to advertise it, but we don't replicate our experiments when we're doing a large screen. Now, when we go back in and after the model makes a prediction that I think you can improve riboflavin by doing this thing, then that's, of course, obviously replicated. But there's a lot of those noisy points, and we know that they're in there, that just over time get averaged out by the model.
Well, thanks.
Everyone should stop doing right. I think that's the message today, is that we should all stop doing right.
James, did you have a question? I saw your hand go up and then it went off.
Yeah, hi, this is Jim Collett, and I, for many years, was working over in Scott's former team. I still in that group doing bioreactor work. And so I'm interested about, one thing is your translation of your work from plates to bioreactors, and then we've used Raman spectroscopy to implement control systems for automatic feeding of, in this case, it was corn stover hydrolysate. And we're able to keep the glucose level between 3 and 5 grams per liter for 48 hours. just on the ramen. So what I'd like to ask is, when you do your measurements of your results and so forth, you're just measuring growth. But as Jeremy was getting back, if you have more of that metabolite information, perhaps on the fly, if you then you could get that, and you can get a faster turnaround than sending everything off to the LC. Having said that, to build a ramen model with the bioreactor, first we needed to implement an auto sampler that would pull a sample every 30 minutes around the clock, for at least 24 hours, and then take that information, take it to the LC, and then get enough to make like a partial least squares predictive model for multiple chemical components. Are you interested in that kind of thing as well, like introducing other channels of measurement that you could potentially use on the fly?
So, yes. So the dynamic models that I showed you, the neural differential equations actually predict the metabolite levels. The predictions are just nonsense because we don't have any data after the initial. It's a little better when you do the perturbations, but we basically view this as being partially observed, where we set up all of the variables in the reactor, and then we only train it based on the growth, based on the optical density.
Mm-hmm.
So if you had metabolite measurements in time series, then you could, in principle, learn those at the same time. And we would like to, we just don't have the measurements. And in terms of turnaround, the best thing you could do to improve learning is do more cycles at a faster turnaround. So if you had 1,000 experiments, the theoretical worst way to do it is do all 1,000 at once and then learn. The theoretical best way is to do one experiment, learn, plan the next one experiment, and do it that way. But for growth assays, that would take 1,000 days, and we can't do that. So the only reason we do batches greater than one is we know it'll be less efficient to learn, but it'll be practically more efficient because we can do it in a batch, and we have to find that right balance. So I think anything that speeds the feedback loop with the data will give you more efficient learning, and that's been well established in the theory for a while. So I'd be all for that.